So this is personalized medicine in its purest form. And the question is a bit, can we really sustain this? Can we really hold up the promise that was made a number of years ago? I mean, it was a very successful concept to, particularly for cancer research. I'm coming from a cancer research center, to increase funding, to make the funders believe in some of the promises that were being made. But I think the time has come that we have to ask some critical question and to answer the question whether we can actually fulfill the promises that were made a while ago. So that's what I wanted to say to start. The second thing is I'm not a geneticist, I'm an epigeneticist. So my personal contribution to personalized medicine as such is a bit limited. Personalized medicine is heavily based on genetics. I mean, one of the key illustrations was to demonstrate that cancer is not caused by single mutations or by two mutations, but actually by a group of mutations. Usually we see a genetic mutation landscape in tumors. So a group of mutations that collaborate to drive cancer, to drive tumor formation. This landscape can be very individual. So almost every patient has a somewhat different mutational landscape, and which is also a very important foundation for the concept of personalized medicine. However, this also has an epigenetic dimension. Similar to the mutation landscape, we also have a epimutation landscape. Like there are genetic mutations that probably most, if not all of you know, there are also epigenetic mutations. Epigenetics is a regulatory layer on the genome that acts on top of genetics. It's usually about regulatory modifications like DNA methylation, chromatin packaging, more about that in a minute. And these modifications can also be altered in cancer. So this would be one of the biomarkers, the various biomarkers that could also be used for patient stratification. And if you have a list at this figure, this was very influential. It's already, almost 20 years old. The red mutations, the red symbols here are the genetic mutations. The green symbols are the epigenetic mutations that were known at the time. The purple markers were the mixed mutations. So it was suggested that epigenetics has a similar impact on tumor formation, a similar functional role like genetics. Now, to explain this a little bit further, what is epigenetics? I will make this short to get to the personalized medicine part soon. Epigenetic mechanisms are very important for cellular differentiation. This is probably the key biological context for which we need epigenetics, at least in humans and in mice. So you can, we have stem cells, and stem cells are pluripotent or even totipotent, meaning they can differentiate in many different specialized cell types. This is always the same genome. Every cell in your body, or most of the cells in your body, have the same genome, but obviously the cell types have different functions. How is this made possible? And the answer to the question is epigenetic programming. We don't access all the parts of our genome in every given cell. Every cell type utilizes specific genes, and the programs that orchestrate this cell type specific use of genes, that's controlled by epigenetic mechanisms. So epigenetic acts during cellular differentiation. Epigenetic programs result in differentiation, but these programs become distorted during tumor formation, resulting in phenotypically more variable cells in the cancer cells. This is the functional contribution that epigenetics might make or makes to tumor formation. One of the key epigenetic modifications is DNA methylation. This is a simple covalent modification of DNA. We have a quarter of our genome is made by cytosine residues. About 4% of these cytosine residues can be methylated, resulting in about 40 million methylation marks in the human genome. And the distribution of these methylation marks is a very important code for epigenetic programming. These methylation marks can instruct gene expression programs, and we can predict whether a gene can be expressed or not based on DNA methylation patterns. And now it has been known for a long time that these DNA methylation patterns that we can really resolve at the single nucleotide levels. We can draw genome-wide maps with DNA methylation patterns, and these methylation patterns can become altered in cancer. This was one. What you see here is one of the first genome-wide analysis where a colon cancer was profiled for DNA methylation and was compared to a normal colonic tissue. And what you see is in this track here is the tumor to normal difference. So we'll see that most of the green bars go to the south, meaning that the tumor has lost methylation to a large extent, but some regions also have gained methylation. This is a typical picture that you see in tumors, a global loss of methylation combined with a gain of methylation at specific genes. This is supposed to underlie the reprogramming of a normal cell towards a tumor state. Obviously, there are some phenotypical differences between tumor cells and normal cells. Normal cells divide in a very controlled cell and they can undergo apoptosis, while tumor cells have an uncontrolled mode of cell division and are often apoptosis resistant. It was for a long time believed that this was underpinned by the epigenetic programs. For example, that if you silence tumor suppressor genes epigenetically, and this is something that has been observed, the cell loses its capacity to control the cell cycle. At the same time, if you silence apoptosis-inducing gene, the cell loses its capacity to undergo apoptosis. And thus, it was suggested epigenetics might confer key attributes to cancer cells. At the same time, it was suggested that we can remedy this, that we can develop therapies to combat this mechanism. This is shown here. This is a classical picture, a promoter of a tumor suppressor gene gaining methylation over time until a sufficient level of methylation was achieved to silence the expression of this gene. And then it was said, let's just target the enzymes that mediate DNA methylation. And this is a DNA methyltransferase and a catalytic domain. It has a nicely defined catalytic pocket. Let's target this catalytic pocket and then reverse epigenetic mutations. Let's inhibit DNA methylation. The cells would go through cell cycle, re-establish the normal methylation pattern, and thereby, if you have the right dose and the right drug, you might reprogram cells from the cancer state to the normal state. This was a nice concept. It was a great idea, but unfortunately it didn't work, at least not the way like it was initially drawn up. There were some problems. For example, some of them are very easy to explain. Epigenetic changes are genome-wide. Only some of these cancer-associated epigenetic changes are genome-wide. Only some of them, however, functionally contribute to the tumor phenotype. If you just use the shotgun to alter epigenetic modification patterns, you do not operate in a very targeted way. You alter the entire epigenome, even though if you might want to only specifically alter some certain genes. So specificity, how do you target specific genes and regulatory regions? This was a major problem. It could never be resolved. Also, the true personalization of this approach was very difficult to achieve. The functional role of epigenetic mechanisms, for example, is surprisingly difficult to prove. You would imagine there's so much literature about this, epigenetic changes associated with tumors. Nevertheless, if you knock down or knock out a DNA methyltransferase gene in the mouse and test its effect on tumor phenotypes, very little has been demonstrated. And also, we don't have enough drug candidates. It's not just DNA methylation, also various histone modifications are altered. Other epigenetic mechanisms exist and become altered in cancer. We don't have drugs to address them all. We don't have the ability to combine these drugs to have a really rational therapy, and which is also due to the fact that drug-drug interactions, a little bit about that in a few minutes, are completely neglected in this approach. If you want a truly personalized therapy, you will have to combine drugs. Those patients have a certain number of mutations. If you have a targeted drug like Gleevec for one mutation and a patient has 2 or 3 or 4 or 10 of these mutations, you would have to administer the equivalent number of drugs. Combining even 2 drugs is a real clinical challenge, and addressing drug-drug interaction in the clinic is a real challenge that has often not been resolved. And if you consider the combinatorial possibilities of, maybe you have 100 drugs available, what goes with what? It's incredibly difficult. And it would have to be addressed before making it available to the patient. So how can we, in the light of these challenges, how can we help the concept? I mean, of course, something is working. I don't want to trash personalized medicine. I mean, a lot has been working over the past 15 years. We have used genome sequencing to establish mutational profiles of individual tumors. We can give a very precise diagnosis now. So how could we make this work? And I think epigenetics can provide a part of the solution, and this is what I would like to explain in the remaining few minutes of my talk. Like I mentioned before, the epigenetic changes that we observe in cancers have most often linked to two things. First of all, cellular transformation. It has been speculated that the epigenetic changes reflect the altered cellular program of cancer cells, that they fuel proliferation, that they fuel apoptosis resistance. And secondly, it has also been said that these epigenetic changes reflect stem cell-like patterns to underpin the tumor stem cell hypothesis. Thirdly, it has also been suggested that the epigenetic patterns that we observe in tumors reflect the tumor cell of origin. That's a very important and very different concept. All tumors originate from a single cell. And methylation patterns in general, if you look in the human genome, they usually reflect cell types. So when I profile the epigenome of a healthy tissue, I can make a clear difference between, neuron, hepatocyte, fibroblast, keratinocyte. This is the main function of DNA methylation. So if I profile any given cell or tissue, I can actually predict the cellular identity of this particular cell type. We have databases for that. There are reference databases for all human cell types. And this is the real power of epigenetics. So if the tumor retains the methylation pattern of the cell of origin, the tumor methylation pattern might actually allow me to identify the cell of origin. This would be very important because right now we are diagnosing cancer by the organ where it has been detected. We talk about brain cancer, liver cancer, colon cancer. However, all these organs are made up of various cell types and various differentiation stages. So some people have believed if you can identify the cell of origin, then you learn a lot about the biological properties of this specific tumor types. And now I will briefly illustrate two examples that have led to this concept. One was we have made some contributions as well, specifically for skin cancer, non-melanoma skin cancer, so white skin cancer and colon cancer. And there are some I think quite insightful results that can illustrate this new development. So we were looking at the DNA methylation patterns of squamous cell carcinoma. This is or cutaneous squamous cell carcinoma. This is the white skin cancer that often occurs with advanced age. It is preceded by a premalignant lesion that is called actinic keratosis. So we obtained a number of samples and analyzed their methylation patterns. I should also say that squamous cell carcinoma has one major advantage, namely that the cell of origin basically is known already. It's the keratinocyte. All squamous cell carcinomas derive from keratinocytes, and this was why this cancer was supposed to be or believed to be very heterogeneous and was not subclassified before. I should also mention that keratinocytes are the end product of a quite sophisticated differentiation process. They result from the differentiation of epidermal stem cells. The human epidermis has a layered pathology or composition. We have the epidermal stem cells in the somewhat deeper layers here, and then differentiation starts, and the more differentiated keratinocytes migrate to the outside until they undergo apoptosis and become, I'm lacking the English word now, become basically released from the skin. When we use array-based methylation profiling of healthy epidermis, actinic keratosis, and squamous cell carcinoma, this is a technology. I should also explain that briefly. We have arrays. We can sequence methylomes. This is possible at single base resolution, like shown before, but we can also use some somewhat simpler array-based technologies where we can analyze a lot of samples in a relatively short period of time. This still allows us to analyze 850,000 of the 40 million methylation mark in the genome, so it gives us reasonable diagnostic power. We use this method for profiling about 50 epidermis samples, healthy, AK, and SCC. And what we observe is, and that was the first interesting observation, that AK and SCC actually show similar methylation patterns. This is perhaps best illustrated here. When we compared, this is a scatter plot. It shows all the methylation probes that were analyzed in AK and healthy. When you compare AK, which is a pre-malignant lesion, to healthy skin, you see a lot of differentiated methylated probes. These are methylation markers that are significantly different between AK and healthy. When you compare SCC to healthy, you also see a lot of methylation differences, more than 300,000 markers. But if you compare SCC to AK, we don't see any significant methylation differences. So it's not the cancer that alters the methylation patterns. It's the transition from healthy to the pre-malignant lesion. This was the first important observation. We already see the cancer-associated markers in the pre-malignant lesion. The second key observation was that we can subclassify both AK and SCC using DNA methylation in two distinct subtypes. These two subtypes emerge in the pre-malignant stage and then are maintained in the cancer. This was observed at keratinocyte, this was observed at enhancer markers, and it turned out, like I said, we can operate with a huge database of reference methylomes from all kinds of cell types, from all kinds of differentiation stages. It turned out that these two subgroups are actually epidermal stem cell-like and differentiated stem cell and differentiated at keratinocyte. So the differentiation stage of the cell type that formed two subtypes of the pre-malignant lesion and then the cancer. What does this mean? This is the model for skin cancer. We have an epidermal stem cell, and for normal epidermal homeostasis, this epidermal stem cell is differentiating into keratinocyte. And then we have two exit stages, basically, where a cell of origin can form to form first a pre-malignant lesion and then later skin cancer. And we can use methylation profiles to subclassify the patient into two biologically different subclasses. This is important because, first of all, you cannot, this was not known before, this kind of subclassification is novel, and also it's biologically important because we interpret the methylation changes that we observe to reflect differences in the cell of origin. The healthy epidermis is a mixture of all kinds of differentiation stages. The tumor is a clonal amplification of the cell of origin. This is why we see the differences, the methylation differences between healthy and disease, because one is a mixture, the one is a clonal amplification. All the time we have been comparing apples with pears, but if you use the matched controls, the right differentiation stage, you will not see any methylation differences anymore. So the old concept of cancer epigenetics using cancer-associated methylation changes for diagnosing cancer does not work here. But a new concept is emerging that you can use the methylation patterns to identify the cell of origin. And if we have different cell types of origin for the same cancer entity, this means that we have novel means for precision diagnosis. A second example, colon cancer. Colon cancer is overall similar in that respect that you have a pre-malignant lesion, the adenomas that develop into carcinomas. Adenomas are completely benign. They can be screened, they can be removed. You have no risk of a relapse. Carcinomas are a cancer. We have about 10 to 15 years from initiation to disease. We have a very high heterogeneity in colon cancers. These were the known challenges. And the questions were, do we have precancers, epigenetic changes at the adenoma stage? And can we use them to subclassify colon cancer? Now I have a problem with the format. Doesn't really matter. We've used the same approach for DNA methylation profiling. We use these arrays. We also, again, accessed the data from the databases. We found 4 subtypes, or 4 epigenetic subtypes of colon cancer. Each of them we could correlate to a specific cell type of origin. And interestingly, the cell types of origin were the stem cells, the transit amplifying cells, and 2 types of differentiated cells. Again, colon cancer, it's known to be a heterogeneous disease. And it is being subclassified using various genetic markers, but there are no real biological explanations for this approach towards subclassification. So you can have a BRAF mutation, you can have a KRAS mutation, but what this means in a broader biological context has never been clear. What we say is we have subtype 1 that is originating from stem cells. We have subtype 2 that is originating from transit amplifying cells. We have subtype 3 and 4 from the more differentiated cells. This can also be correlated to the clinical prognosis. Unfortunately, this is a bit out of order, but maybe I'll come back to that. I don't know what I'm missing with the slides that I can't show. So let me get to another thing. So 4 subtypes of colon cancer. Interestingly, 3, we can also recapitulate this when we use colonic crypts. Use laser microdissection to obtain the cells from the base, the neck, and the top of the crypt. We see the same methylation patterns like that we saw in the subtypes of colon cancer. We can also use adenomas that have no malignant potential. These adenomas have the same methylation subtypes like the colon cancer, which again means that the methylation pattern from the cell of origin, from the crypt, is maintained to the benign state, the adenoma, the precursor stage, and then to the carcinoma. So cell of origin methylation signatures are maintained during colorectal carcinogenesis, and again, this is I'm not sure what happened there, but I sent the slides in advance. What this slide is supposed to show is that we even see an effect on our subclassification gene on the clinical prognosis. For example, we know that those patients who have the stem cell subtype have a much poorer outcome, much more aggressive tumors. This is a clinical significant and potentially highly relevant observation, so we actually suggest to profile these patients for the methylation subtype in order to predict the clinical outcome. And also potentially, this would also be very important to make recommendations for the treatment. You can imagine if a tumor originates from a stem cell or from an intermediate cell or from a differentiated cell, it has a different metabolism, it has a different cellular makeup. So this needs different therapies, and to make these kinds of predictions could potentially be quite helpful for therapy. So what we essentially, now I'm coming to the end and hopefully have some good slides here, what I essentially propose is to use the power of DNA methylation pattern in order to improve the level of resolution for cancer diagnosis. Traditional diagnosis either primarily uses the organ of the primary tumor for classification. Do we have liver cancer, brain cancer, or colon cancer, and then uses genetic markers in order to somewhat arbitrarily subclassify these cancers. What we propose is use DNA methylation profiles in order to identify the cell type of origin and the differentiation stage for the cell type of origin for every tumor. This should work for every tumor. We have shown it for skin cancer and colon cancer. Others have shown it for leukemias and brain cancer. This is something that is really gaining a lot of traction right now. And what we propose, nothing less than changing the tumor classification system from the organ to the cell type of origin. So what does this mean for personalized oncology? This is sort of like a personal summary from my point of view. Initially, I was very excited when personalized oncology was first suggested. It made a lot of sense. But after almost 15 years now, I think it's time to say that some of the initial promises of personalized diagnosis and treatment were not fulfilled. It works very well on the diagnostic side. This is something where we could keep the promises due to the increasing availability of whole genome, methylome, transcriptome sequencing. But whether this helps the patient or not is still an open question. There are some beautiful cases where the power of personalized medicine could be proven. I mean, one key example is still Gleevec. You have this BCR-ABL fusion. You get Gleevec and you get a lot of clinical benefit. But I'm talking about this. These are small groups of patients. The big killers, colon cancer, lung cancer, they do not really benefit from personalized therapies right now. And I don't think that any major changes are on the horizon. Drug development is extremely expensive. If we need an individualized drug for every mutation, I don't think we can afford it. A phase 1 candidate in oncology costs $50 million, and a phase 1 candidate is still, a drug candidate that has a 10% chance of being clinically effective. This kind of money is simply not available. And particularly if in the end we are talking about drugs that might only be beneficial for a very small group of patients. Drug-drug interactions remain a major unresolved obstacle. Few people want to touch this. Few people want to discuss this. But in the end, personalized medicine will only work in cancer if you are able to combine drugs, not just 2, even 3 or 4. This is a tremendous pharmacological challenge that has not been met. Also, and this is the last major challenge, also not really often talked about, but key tenets of personalized oncology are being challenged now, the driver concept. Personalized medicine is built on the assumption that tumors are driven by certain genetic mutations that affect factors important for cell division or the control of cell division, the driver mutations. But more recent research that was done with biopsies, multiple biopsies from individual tumors, shows that at least some of the tumors, if not the majority, are evolving neutrally so that they are not driven by specific mutations. This is not well understood, but it's a huge discussion going on now in cancer genetics, and it remains to be addressed. If this is true, it would really rock the conceptual foundation of personalized oncology. So what I'm saying is, in principle, the proof of concept for personalized oncology remains to be established for the majority of cancer patients. However, not all is bad. Major advantages were made with respect to diagnosis. What we can do now, what we can claim, is that we are able to make precision diagnosis at the genetic level. The mutations, we can map them all if we sequence the genome. We can make a very precise diagnosis. So what I propose is that we should get away from this grand design, this one. I don't think we can fulfill this promise, but what we can say, what we should use, is the concept of precision oncology. Make the diagnosis as precise as possible, make the treatment as precise as possible, but know the limitations. We can't make it personal, we can't really individualize it, but we can make it precise. Okay, I think I will show one more slide. The work in my group was not done by me, but by Manuel and Felix here. We collaborate also with Judith Bergman in this concept in the framework of the Helmholtz-Israel collaboration or initiative in personalized medicine. And a major contributor was also Heinz Linhardt, an oncologist working on colon cancer and skin cancer. Thank you for your attention. I'm looking forward to answering questions.
Thank you very much. Do we have any questions? Please. Yes.
Thank you, Frank. It was really fascinating.
I get closer to you. I'm hard of hearing, which is why I like to be close to the
I'm not sure this works, does it?
Yeah.
That works better. Yeah, it was supposed to work, but I'm not commenting on this great idea of cell of origin. I think it sounds very convincing to me, but I'm not in the field. I have a problem, or let's say a question concerning epigenetics. Can you really distinguish so well between genetics and epigenetics? We had this discussion. Because epigenetics is, in a way, a result of a genetic event. I mean, the repressors which recruit the methylases are DNA specific. And shouldn't there be, for any epigenetic change, also a genetic change? Like Adrian Burke has shown for autism, that the changes of methylation are results of mutations. So
I'm not sure about frankly, I'm not sure whether I can agree with the statement that epigenetics is underpinned by genetics. This question was very important for addressing the possibility that epigenetic modification patterns are influenced by environmental factors. In that case, we usually resort or fall back to the issue that we always have genetic changes underpinning the epigenetic operation. However, for a process like cellular differentiation, I'm sure that there are no genetic changes that are associated with the epigenetic changes. This is always the same genome that proceeds from a stem cell to a transit amplifying cell to a differentiated cell. There are no genetic changes involved in this process. So this is what I'm trying to transport into the concept of the cell of origin and to utilize for this.
No, not mutations, but it's a genetic case, but they are genetically regulated. I mean, there are always repressors which recruit the methylases. And the repressors are brought by our DNA sequence specific, and the methylases are not. So the differentiation must be regulated. It's not a random distribution of methyl groups. So it is a genomic regulation. It's not by new mutations, of course. Yeah.
This could certainly be, an additional factor that influences the cancer genome. But I found it quite particularly surprising that we got the results that we got using, there's always filtering involved in this kind of bioinformatic analysis, but the filtering that we did was to use the most differentially methylated hosts. Those shed light on the differentiation stage.
Oh, that's great. Not contradicting that.
My question comes from the other side. Mechanisms which epigenes the genome.
Would you speak up, please?
I don't know. The mechanisms through which the epigenome works mostly is the transcriptome. So have you compared using your classification based on epigenome to the transcriptome, combining genetics and transcriptome? By and large, you expect that those would be quite similar.
Yeah, before we have the technologies to analyze epigenomes, the technologies to analyze transcriptomes were already available and quite refined. Of course, cancer subclassification by transcriptomics has been done and used and has been successful to some extent. Colon cancer is actually a good example. There were several studies that subclassified colon cancer according to the gene expression profiles and actually already found a link to the cell of origin. But the differences and the relationships were not as clear as we observe them now with methylation. I believe that the methylation patterns are more resistant to environmental influences, more stable and more stably maintained through the process of tumorigenesis than the transcription profiles. And I don't think that we have a perfect link between DNA methylation and transcription. I mean, some methylation events and effects can predict transcriptional changes, but you cannot really predict the overall transcriptome from the methylome. This is still not feasible. Probably will never be feasible. But still, it is a signature that is maintained from the cell of origin, obviously quite stably. So I wouldn't necessarily the cancer phenotype later can be influenced by a lot of factors. But knowing this program and knowing the cell of origin this is what I'm saying, would be a major advantage just for tumor cell classification.
I think in OncoTrack we found quite a lot of power into transcriptome, and we did not see as much edit information in the epigenome, but that is obviously on a case-by-case basis. Generally, I would argue personalized medicine has not failed, it hasn't started yet.
No, I mean,
I should
even
keep
more
mentioning.
So colon cancer is again a good example. I mean, there's currently something called the consensus molecular subtype system, CMS system, which is the most advanced molecular system for the subclassification of colon cancer. This is based on the analysis of gene expression patterns. Again, they see 4 subtypes are observed. And we see a substantial overlap between the methylation subtypes and the expression subtypes and the CNS subtypes. Nevertheless, we have a lot less gray area. The expression patterns leave a lot of unresolved cases that we don't see with the methylation profiles. Also, the clinical differences, the prognostic power of the marker is higher for methylation than for gene expression.
And in general, I think that much of the discussion on personalized medicine seems to be that the Man on the Moon program is declared failed before the first rockets have been built.
Thank you. I have one question, more of the bigger picture and the link to the personalized medicine, because I think it's interesting that one of your approaches, saying the profiling of the cell of origin that improves subclassification is quite contradictory to what the approach of personalized medicine in the beginning was, namely to kind of say, well, we can go without all of the tumor classification if we understand the molecular making up of any single cells. We can kind of have a follow-on tumor agnostic approach, and you are going quite in a different direction.
That's a good point. I mean, I think first of all, it would be important, and that would probably be the most feasible part to see whether the methylation pattern, the cell of origin classification, can predict the response to available drugs in a better way than other markers. So that would be my assumption. I'm assuming that it may work. I'm assuming that a stem cell, if a tumor is formed by a stem cell, it has a different molecular, metabolic makeup than a tumor that is formed by differentiated cells. Maybe we can exploit this. But I wouldn't go so far as to say that we should make targeted drugs for every differentiation stage, because again, I don't think it's really feasible. I don't think it's affordable. But it would certainly be interesting to check the ability of these markers to predict the response to existing drugs.
Any more questions? I have one question. If we have to decide, like in your opinion, like it's hypothetical, between what the epigenetic marks are telling us we should treat the patient versus the genetic mutations are telling us that we should treat the patient, how can we define the hierarchy or how to take a decision when you cannot combine drugs or you cannot combine the treatment? How can we start?
So if you have a conflicting advice
exactly,
from
because nothing is that simple in biology.
That's again a very good question. I don't know. I mean, I think we have to try. Both, and also, don't get me wrong, I'm not saying we should not use the power of the genetic information that we have. Maybe we can use this as an additional layer, but I would like to connect this to Eva's question. If we can see that the epigenetic markers predict patient response to certain drugs in a better way than genetic markers, then we should favor the epigenetic markers. If the analysis of this kind of data shows that the genetic markers work better, then we should stick to the genetic marker. If we are talking about a drug, a targeted drug that hits a specific mutation, of course I wouldn't care about the epigenetic marker. The BCR-ABL mutant patients should still be treated with Gleevec no matter what the methylation profile is. But this is only beneficial, this kind of approach, for maybe 5% of the patient. I'm talking about 95% of the other patients that do not currently benefit from this kind of treatment. And for those, maybe the epigenetic markers may be of greater help than the genetic marker, but time will tell. The analysis need to be done.
Any more questions? If not, we'll go for a coffee break, and thank you very much for your time.